Biomolecules, worksheet part 1. Trichloroacetic acid. Acid to analyze the chemical composition. Trichloroacetic acid. Acid to analyze chemical composition. Filtrate. Acid soluble pole. Retentate. Acid insoluble pole. Filtrate. Acid soluble pole. Retentate. Acid insoluble pole. Molecular weight less than 1000 Dalton. Biomicromolecule. Molecular weight less than 1000 Dalton. Biomicromolecule. Molecular weight more than 1000 Dalton. Biomacromolecule. Molecular weight more than 1000 Dalton. Biomacromolecule. Represent cytoplasmic composition. Represent cytoplasmic composition. Filtrate. Represent cytoplasmic composition, filtrate. Contain molecule from cytoplasm and organelle, retentate. Contain molecules from cytoplasm and organelles, retentate. Dried living tissue burnt into ash. It is for the separation of inorganic elements. Dried living tissue burnt into ash. It is for the separation of inorganic elements. Inorganic elements in ash, calcium, magnesium. Inorganic elements in ash, calcium, magnesium. Inorganic compounds in ash, sulfate and phosphate. Inorganic compounds in ash, sulfates and phosphate. Amino acid, lipid, sugar, nitrogen bases. Amino acid, lipid, sugar, nitrogen bases are called biomicromolecule. Proteins, polysaccharide, nucleic acid, biomacromolecule. Proteins, polysaccharide, nucleic acid are called biomacromolecule. Acid soluble fraction contain biomicromolecule. Acid soluble fraction contain biomicromolecule. Acid insoluble fraction contain biomacromolecule. Acid soluble fraction contain biomicromolecule. Acid insoluble fraction contain biomacromolecule. Biomicromolecules of acid insoluble fraction, lipids. Biomicromolecules of acid insoluble fraction, lipids. Alpha carbon, amino group and base group on same carbon. Alpha carbon, amino group and base group on same carbon. Carboxyl group attached to an R group. Carboxyl group attached to an R group, lipids. Carboxyl group attached to an R group, lipids. Substituents of methane, amino acid. Substituents of methane, amino acid. R group can be methyl, ethyl, or CH2 with a higher number. Lipids. R group can be methyl, ethyl, or CH2 with higher number. Lipids. 4 substituent groups occupy 4 valency positions. Amino acid. 4 substituents occupy 4 valency position. Amino acid. Number of carbons in palmitic acid, 16. Number of carbons in arachidonic acid, 20. Number of carbons in palmitic acid, 16. Number of carbons in arachidonic acid, 20. Hydrogen, carboxyl group, amino group, and variable R group, amino acid. Hydrogen, carboxyl group, amino group, and a variable group, amino acid. R group with hydrogen, glycine. R group with methyl, alanine. R group with hydroxymethyl, serine. R with hydrogen, an amino acid. R group is with hydrogen, glycine. R group with methyl, alanine. R group is replaced by hydroxymethyl, serine. If amino groups are more in amino acid, it is base. If carboxyl groups are more in amino acid, it is acidic. If amino groups are more in amino acid, it is base. If carboxyl groups are more in amino acid, it is acidic. Acidic amino acid, glutamic acid. Acidic amino acid, glutamic acid. Base amino acid, lysine. Base amino acid, lysine. Neutral amino acid, valine. Neutral amino acid, valine. Acidic, glutamic acid, base, lysine, neutral, valine. Aromatic amino acid, tyrosine, phenylalanine, tryptophan, histidine. Aromatic amino acid, tyrosine, phenylalanine, tryptophan, histidine. Fatty acids without double bond, saturated. Fatty acids without double bond, saturated. Fatty acids with one or more carbon double bonds, unsaturated. With the fatty acid with one or more carbon double bonds, unsaturated. Trihydroxypropane, glycerol. Trihydroxypropane, glycerol. One fatty acid esterified with glycerol, monoglyceride. Two fatty acid esterified with glycerol, diglyceride. Three fatty acids esterified with glycerol, triglyceride. Oils which have lower melting point, gingerly oil. Example, gingerly oil. Oils have lower melting point. Example, gingerly oil. Lipids with phosphorus and phosphorylated organic compound, phospholipid. Lipids with phosphorus and phosphorylated organic compound, phospholipid. Sugar plus base, nucleoside. Sugar base phosphate, nucleotide. Sugar plus base, nucleoside. Sugar plus base plus phosphate, nucleotide. Adenosine, guanosine. Adenosine, guanosine. Adenine, guanine are nucleosides. So, adenosine, guanosine, nucleoside we call purines. Thymidine, cytidine, uridine, nucleoside, pyrimidine. Thymidine, cytidine, uridine, nucleoside, pyrimidine. Adenilic acid, guanilic acid, nucleotide. It includes sugar base phosphate. Adenilic acid, guanilic acid, nucleotide, purine. Thymidilic acid, cytidilic acid, uridylic acid, nucleotide, pyrimidines. So purines, adenine and guanine, pyrimidines, thymine, cytosine, uracil. So when they combine with the sugar and phosphate, what do you say? Sugar and base, nucleoside. Sugar, base, and phosphate, nucleotide. Individually, adenine plus sugar, adenosine. Guanine plus sugar, guanosine. Thymine plus sugar, thymidine. Cytosine plus sugar, cytidine. Uracil plus sugar, uridine. Adenine plus sugar and phosphate, nucleotide, adenylic acid. Guanine plus sugar plus phosphate, guanylic acid. Thymine plus sugar plus phosphate, thymidilic acid. Cytosine plus sugar plus phosphate, cytidilic acid. Uracil plus sugar plus phosphate, uridylic acid. Let me say once again, sugar plus base, nucleoside. Sugar base phosphate, nucleotide. Adenosine, guanosine, thymidine, cytidine, uridine are nucleosides. Adenylic acid, guanylic acid, thymidilic acid, cytidilic acid, uridylic acid are nucleotides. Adenine, guanine, purine. Thymine, cytosine, uracil, pyrimidine. Okay. Adenosine, guanosine, nucleoside, purine it is. Thymidine, cytidine, uridine, nucleoside, pyrimidines they are. Adenylic acid, guanylic acid, nucleotide, purine. Thymidilic acid, cytidilic acid, uridylic acid, nucleotide, pyrimidine. Lipids, fats, and oils based on. Lipids are classified into fats and oils based on melting point. Lipids with lower melting point, oils. Lipids found in cell membrane, phospholipids. Lipids found in cell membrane, phospholipids. Carbon compounds with heterocyclic rings. Carbon compounds with heterocyclic ring, nitrogenous bases, adenine, thymine, guanine, cytosine, uracil, are the carbon compounds with heterocyclic ring. Example for phospholipid, lecithin. Example for phospholipid, lecithin. Phosphate group esterified with sugar of nucleoside. Phosphate group esterified with sugar of nucleoside, nucleotide. Phosphate group esterified with sugar of nucleoside, nucleotide. Alkaloid, flavonoids, rubber, essential oil, antibiotic, colored pigment, scents, gum, spices. All these are called secondary metabolites. Alkaloid, flavonoids, rubber, essential oil, antibiotics, colored pigment, scents, gum, spices are called secondary metabolites. Sugars, amino acid, lipids, nucleic acid. Primary metabolites. Sugars, amino acid, lipids, nucleic acids are called primary metabolites. Molecular weights ranging between 18 to 800 Dalton approximately. Biomicromolecule acid soluble pool. Molecular weight ranging between 18 to 800 Dalton approximately. Biomicromolecule acid soluble pool. Organic compounds of acid insoluble fraction. Proteins, polysaccharides, proteins, polysaccharides, and nucleic acid, lipids. Organic compounds of acid insoluble fraction. Proteins, polysaccharides, nucleic acids, and lipids. Proteins, polysaccharide, nucleic acid, lipid. Cell membranes and other membranes broken into pieces while grinding and form vesicles. Cell membranes and other membranes broken into pieces while grinding and form vesicles, which it include lipids. Linear chains of amino acids linked by peptide bonds. Linear chains of amino acids linked by peptide bonds. Protein. Polymer of amino acid. Protein. One type of monomer repeating n number of times. Homopolymer. One type of monomer repeating n number of time. What do you say? Homopolymer. Protein is heteropolymer. Protein is heteropolymer. Amino acid which human body cannot synthesize. Essential amino acid. Amino acid which human body can synthesize. Non-essential amino acid. Most abundant protein in animal world, collagen. Most abundant protein in animal world, collagen. Most abundant protein in animal world, collagen. Ribulose biphosphate carboxylase oxygenase, Rubisco. Most abundant protein in the world. Ribulose biphosphate carboxylase oxygenase, Rubisco. Most abundant protein in the world. Long chain of sugars, polysaccharide. Long chain of sugar, polysaccharide. Homopolymer carbohydrate, cellulose. Homopolymer carbohydrate, cellulose. Storehouse of energy in plant tissues, starch. Polymer of fructose, inulin. Polymer of fructose, inulin. Storehouse of energy in animal tissues, glycogen. Right end of glycogen chain, reducing end. Right end of glycogen chain, reducing end. Left end of glycogen chain, non-reducing end. Left end of glycogen chain, non-reducing end. Branches in the form of carbon, polysaccharide. Branches in the form of carbon, polysaccharide. Helical secondary structure, starch. Helical secondary structure, starch. Does not contain complex helices, hence cannot hold iodine. No blue color. Does not contain complex helices, hence cannot hold iodine. No blue color appear. Starch iodine combination, blue color appear. Starch iodine combination, blue color appear. Plant cell walls are made of cellulose. Plant cell walls are made of cellulose. Glucosamine N-acetyl galactosamine. Glucosamine N-acetyl galactosamine. Amino sugars. Glucosamine and N-acetyl galactosamine. Amino sugar. Paper made from plant pulp and cotton fiber contain cellulose. Paper made from plant pulp and cotton fiber, cellulose. Complex polysaccharide of exoskeleton of arthropod, chitin. Complex polysaccharide of exoskeleton of arthropod, chitin. Can hold iodine molecule in the helical portion, starch. Can hold iodine molecule in the helical portion, starch. Hence it absorbs iodine, it appear blue color. Most complex polysaccharides are homopolymer or heteropolymer. Homopolymer. Most complex polysaccharides are homopolymer. Building block of nucleic acid, nucleotides. Building block of nucleic acid, nucleotide. Heterocyclic compounds of nucleic acid, nitrogenous bases. Monosaccharide of nucleic acid, ribose sugar. Monosaccharide of nucleic acid, ribose sugar. Phosphoric acid of nucleic acid, phosphate. Phosphoric acid of nucleic acid, phosphate. Skeletal heterocyclic rings of nucleic acid, purine and pyrimidine. Skeletal heterocyclic rings of nucleic acid, purines and pyrimidine. Pentose or deoxyribose. Pentose or 2-deoxyribose, ribose sugar. Pentose or 2-deoxyribose, ribose sugar. Nucleic acid with deoxyribose, DNA. Nucleic acid with ribose, RNA. Nucleic acid with deoxyribose sugar, DNA. Nucleic acid with ribose, RNA. Inorganic chemistry structure invariably refers to the molecular formula. We have an example, sodium chloride and magnesium chloride. In organic chemistry, two-dimensional view, molecular structure, we have example, benzene and naphthalene. Thank you.